Bismillahirrahmanirrahim. Dear students, Assalamu alaikum and welcome to office.com.pk. Dear students, this is your own platform where you come and learn, and we try to provide you with the best of knowledge in the best possible way we can. If you really like our videos, please do not forget to subscribe our channel and also hit the bell icon so that whenever we upload a new video, you can get it. And also share your views in the comment section below. Your positive comments will help us boost our morale, and your negative comments will help us improve the way we are doing. Also share the channel to your fellows and friends so that more and more people can benefit from the knowledge that is being shared on the channel. So this was a little request. Previously we were discussing about how DNA encodes protein structure, how cell uses RNA to make proteins, and we also studied the types of RNA in the previous videos. Today we are going to discuss the very important process of transcription, DNA transcription, or you can say it is DNA transcribing. Basically, this is the first step of gene expression. Gene expression is divided into two steps. I told you in the previous video that it is divided into two steps. The first one is transcription and the second one is translation. Gene expression has two steps: transcription and translation. Now transcription is our topic of today. We will discuss translation later. Now what is DNA transcription? Basically DNA transcription is formation of an mRNA copy of DNA. What does it mean? From DNA, messenger RNA is synthesized in the presence of an enzyme which is known as RNA polymerase enzyme. RNA polymerase enzyme. This RNA polymerase enzyme is responsible for copying the information from DNA to messenger RNA. This is the first step of gene expression. Now before we move towards the actual process, let me tell you that the part of DNA or that section of DNA which participates in the process is known as transcription unit. Transcription unit. The whole process is completed with the help of different enzymes, but the part of DNA which is taking part in this whole transcription process is known as transcription unit. The transcription unit further consists of three parts. The first one is promoter site. The second one is the gene sequence, and the third one is stop sequence. Now if we look closely, at a DNA molecule, this is the double helical structure. Now let us suppose that DNA is transcribed or transcripted at this site. So this will be the gene sequence. Now the site which is present before where the transcription will start is the promoter site. This is the promoter site, and the point where the DNA transcribing or DNA transcription will end will be the stop sequence. Just like DNA replication, the DNA transcription also occurs in 5' to 3' direction. It means that this is the direction of newly synthesized messenger RNA. The newly synthesized RNA which will be formed will be in the direction of 5' to 3'. It means that if we want to synthesize this messenger RNA, it will be synthesized against 3' to 5' of the DNA. Now you have to keep another thing in your mind, that if we want to copy this any unit, any DNA sequence on this strand, then we will use the opposite strand to make the messenger RNA copy, because we need this sequence. For example, here we have CAT sequence and the opposite will be GTA, and we want CAT sequence, but we will use the GTA sequence. Our messenger RNA will be formed against this strand, and ultimately the new strand will have CAU, because in RNA we have uracil instead of thymine. So this CAU will be transmitted to ribosomes. This CAU will be just like CAT. That will be our required DNA sequence. Now, what are the requirements? The requirements are enzymes and nucleotides. The enzymes are RNA polymerase. When we were copying DNA from DNA, we used DNA polymerase. Now we are copying RNA from a DNA, then we will use RNA polymerase, and similarly we will use helicase that will unwind the whole structure of this DNA, and the nucleotides will be of RNA. The RNA nucleotides will be used, that will be added to the newly growing strand of the messenger RNA. While we were studying the DNA replication, we studied about the replication fork. In the same way, when DNA transcription starts, then there is the formation of transcription bubble. The transcription bubble will indicate the presence of RNA polymerase, this helicase enzyme, and all these sites of DNA transcription, and the function of all the components.